In this activity, you will examine the external features of eudicot leaves. Unlike monocot leaves, which tend to have a fairly uniform structure, eudicot leaves show tremendous diversity in shape, size, margin type, venation pattern, and arrangement on the stem. Your goal is to learn how to recognize these features and use them to classify leaves based on venation, leaf margins, phyllotaxy, and whether a leaf is simple or compound. A typical eudicot leaf consists of a flattened blade or lamina where most photosynthesis occurs. The lamina is attached to the stem by a petiole, which positions the leaf for optimal light capture. Some eudicots also have stipules, small leaf-like structures at the base of the petiole, although these are not present in all species. In the axil between the petiole and the stem, you will find an axillary bud, an important feature for distinguishing true leaves from leaflets. One defining feature of eudicot leaves is reticulate venation, a branching net-like vein pattern. The benefit of net-like reticulate venation is that it allows for more efficient coverage and redundancy in nutrient and water transport. If part of the leaf is damaged, alternative pathways can still carry resources enhancing survival. There are two common types. In pinnate venation on the left, a single main midvein runs through this leaf with smaller veins branching off like ribs of a feather. You can see an example of this in the leaves of pigeon pea. In palmate venation on the right, several major veins arise from a single point at the base of the leaf and spread outward, resembling the fingers of a hand. You can see examples of this with fig and papaya. Both patterns provide multiple pathways for water and sugar transport and add mechanical support to the leaf blade. Eudicot leaves also differ in the shape of their edges, known as leaf margins. An entire margin is smooth with no teeth or indentations. A serrate or toothed margin has sharp tooth-like projections. A lobed margin has deep indentations that divide the blade into distinct lobes, as seen in many oak and maple leaves. In a parted leaf, the margin has an indentation or indentations that go more than halfway to the leaf midrib or midline. These are just the main categories. There are a variety of leaf margins in between these categories. You will classify each specimen you observe using these margin categories. The arrangement of leaves on the stem is called phyllotaxy. In an alternate arrangement, one leaf occurs per node, alternating sides along the stem. In an opposite arrangement, two leaves arise at the same node or on opposite sides. In a whorled arrangement, there are 3 or more leaves emerge from a single node. Phyllotaxy is often consistent within plant families and can be a useful identification tool. Leaves may be simple, consisting of a single undivided lamina, or compound divided into multiple leaflets. In palmately compound leaves, all leaflets arise from one point. In pinnately compound leaves, leaflets are attached along a central axis called the rachis. There are also bipinnate and even tripinnate leaves. Remember, the axillary bud occurs at the base of the entire leaf, not at the base of individual leaflets. This feature allows you to distinguish a compound leaf from a branch with multiple simple leaves. Before making your observations, pause and consider this question. Why do eudicot leaves often have net-like venation, and how does this benefit the plant in terms of support, transport, and resistance to damage? Consider the effectiveness of coverage along the leaf by having a branching network and how alternative water and nutrient pathways may enhance survival if the leaf is damaged. Write your prediction in your lab notebook, then continue. Let's practice classifying some plants before you have to do it on your own. We'll start with the sea grape on the left. You can see the main stem running down the middle with the leaves attached. Do you see any petioles? You shouldn't, because those leaves are attached directly to the stem. That type of attachment is called sessile. Now look at the veins. What type of venation do you see? Parallel, palmately reticulate, or pinnately reticulate? Notice that there is a main midrib with smaller veins branching off from it. That tells us this leaf has pinnately reticulate venation. Next, look at the edge of the leaf. What type of margin is it? Entire, serrate, lobed, or parted? Since the edge is smooth with no teeth or deep indentations, it has an entire margin. Now let's look at how the leaves are arranged on the stem. Is the phyllotaxy opposite, alternate, or whorled? You only see one leaf per node, so this is an alternate arrangement. Finally, is this a simple or compound leaf? You're looking at one continuous undivided blade or lamina, so this is a simple leaf. Now let's switch to the Pride of Barbados on the right. First, what is the leaf arrangement or phyllotaxy on this plant? At first glance, you might think the leaves are opposite if you focus on the small leaflets. But remember, we need to look at the main stem, which is right here. Once you find the stem, you can see that there is only one leaf per node, which means the arrangement is alternate. Next, is this a simple or compound leaf? This is definitely a compound leaf. Now the question is, what type of compound leaf? Palmate, pinnate, or bipinnate? You can see a central rachis, but instead of individual leaflets attaching directly to it, the leaf divides again into smaller leaflets. That tells us this is a bipinnate compound leaf. On the left we have firebush, and on the right we have pigeon pea. Let's start with the firebush. What type of phyllotaxy do you think it has? If you said opposite, take a closer look. You'll notice that in many places there are actually 3 leaves attached at the same node. When you have 3 or more leaves at a node, the arrangement is called whorled. Now look at the pigeon pea. How many leaves do you see at each node along the stem? You only see 1 leaf per node, so this has an alternate phyllotaxy. Next, let's decide whether the leaves are simple or compound. The firebush leaf is a single undivided blade, so it has a simple leaf. The pigeon pea, on the other hand, has a leaf that is divided into separate leaflets. You can clearly see 3 leaflets attached along a central axis, which means it is a pinnately compound leaf. Let's reinforce some of these concepts. First, you will draw a eudicot leaf. At this point, you don't need to classify it. Be sure to label the lamina, the petiole, and at least one vein showing the reticulate pattern. Focus on accuracy of structure rather than artistic detail. Pause the video to make your drawing. On the next few slides, you will examine several eudicot specimens. Draw and fill out this table in your lab notebook. For each specimen, record whether the leaf is simple or compound, the type of compound leaf, if applicable, the margin type, the venation pattern, and the phyllotaxy. Pause the video to draw the table. Your first specimen is Shumard oak, Quercus shumardii. Take a look at the stem in the center with its large buds in the axils and determine if it is simple or compound. If it is compound, what type? Palmate, pinnate, or bipinnate? What is the type of leaf margin? Entire, serrate, lobed, or parted? Does this have pinnate, palmate, or parallel venation? Does it have alternate, opposite, or whorled phyllotaxy? Note each of these in the table in your lab notebook. Your second specimen is pink trumpet tree, Tabebuia rosea. Take a look at the brown stem in the center. Attached to the stem is a green rachis with 5 leaflets attached at one point. Determine if it is simple or compound. If it is compound, what type? Palmate, pinnate, or bipinnate? What is the type of leaf margin? Entire, serrate, lobed, or parted? Does it have pinnate or palmate or parallel venation? Does it have alternate, opposite, or whorled phyllotaxy? Again, focus on the stem to determine the phyllotaxy. Note each of these in the table in your lab notebook. Your third specimen is Cuban oregano, Coleus amboinicus. Take a look at the stem in the center and determine if it is simple or compound. If it is compound, what type? Palmate, pinnate, or bipinnate? What is the type of leaf margin? Entire, serrate, lobed, or parted? You can get more specific on this one if you want by going back and looking at the leaf margin slide, since this one is in between. Does it have pinnate, palmate, or parallel venation? Does it have alternate, opposite, or world phyllotaxy? Note each of these in the table in your lab notebook. Your fourth specimen is live oak, Quercus virginiana. Take a look at the stem in the center with its large buds in the axils and determine if it is simple or compound. If it is compound, what type? Palmate, pinnate, or bipinnate? If it is, what is the type of leaf margin? Entire, serrate, lobed, or parted? Does it have pinnate, palmate, or parallel venation? Does it have alternate, opposite, or world phyllotaxy? Note each of these in the table in your lab notebook. Your last specimen is hibiscus, Hibiscus rosa sinensis. Take a look at the stem in the center and determine if it is simple or compound. If it is compound, what type? Palmate, pinnate, or bipinnate? What is the type of leaf margin? Entire, serrate, lobed, or parted? Does it have pinnate, palmate, or parallel venation? Does it have alternate, opposite, or world phyllotaxy? Note each of these in the table in your lab notebook. Now let's think more deeply about what you've observed. For the first question, focus on the axillary bud. Ask yourself, where do you see buds forming? At the base of the entire structure or at the base of each individual leaflet? Remember that true leaves, whether simple or compound, have only one axillary bud at their point of attachment to the stem. For the second question, compare leaves with pinnate versus palmate venation. Look at the overall outline of the blade and how the major veins support that shape. Consider whether the vein pattern seems to reinforce the way the leaf is expanded or divided. For the third question, think like a plant taxonomist. Which specimens share the most traits in common, such as venation type, margin, phyllotaxy, and simple versus compound structure? Then ask yourself whether similarity in multiple independent features might suggest shared ancestry rather than coincidence. Pause the video and write your responses in your lab notebook using your observations to support each explanation. While leaf shape is often used to identify plant species, some plants produce more than one leaf form on the same individual. This phenomenon is called heterophylly, which literally means different leaves. The plants shown here, cotton, cilantro, larkspur, buttercup, and jatropha, are classic examples. In cotton, young leaves may be relatively smooth or only slightly lobed, while mature leaves often become deeply lobed. In cilantro, the lower leaves are broad and rounded with shallow lobes, whereas the upper leaves are finely divided and feathery. Larkspur show a similar pattern with broader, less divided leaves near the base and increasingly dissected leaves higher on the stem. Buttercup species can display dramatic differences, especially when growing in different environments. Submerged leaves are often thin and highly divided, while aerial leaves are broader and lobed. Jatropha may produce entire, shallowly lobed or deeply lobed leaves on the same plant, depending on age and growing conditions. In all of these cases, the plant's genetic identity remains the same, but leaf shape changes with developmental stage, position on the stem, or environmental conditions such as light and water availability. These examples remind us that while features like venation and attachment are consistent with major plant groups, the overall outline of the leaf blade can be highly plastic and responsive to the plant's environment. To summarize this activity, Eudicot leaves displays a wide range of forms, but they share several unifying structural features. Their net-like or reticulate venation creates multiple pathways for water and sugar transport and helps support broad, flattened blades. The presence of a petiole and often stipules, along with the position of the axillary bud, allows you to distinguish simple leaves from compound leaves and leaflets from branches. Patterns of leaf arrangement on the stem, alternate, opposite, or whorled, are consistent within species and provide valuable clues for plant identification. Overall, the external features you observe demonstrate how leaf structure is closely tied to function and how shared traits can reflect evolutionary relationships among eudicot plants.